I was invited to give a talk on the tailored treatments in ovarian cancer from basic research to clinical practice. And I thought that it was really a good opportunity to discuss together which are the good preclinical data which justify the clinical developments of new drugs in ovarian cancer, and also to discuss the study design through which they were brought into the clinics. And I thought that's really in ovarian cancer there are two new important classes of drugs, which are the PARP inhibitors and immunotherapy. And I've started talking of the PARP inhibitors because really they've changed the treatment in ovarian cancer, also because their use is based on strong biological data because of the relevance of BRCA in the development of high-grade serous ovarian cancer, which represent more than 70% of the advanced stages of ovarian cancer, and because also BRCA is not an isolated gene, but it's part of the big family of genes, which is called homologous recombination system, and which is very important for repairing the DNA damage caused by the most active agents in ovarian cancer, which are platinum compounds, alkylating agents, and so on. So just to have a dysfunction in this family of genes, which are responsible of repairing the DNA after the damage, means that the tumor in some way remains always sensitive to platinum or to other cytotoxic treatment. Because there's no way to repair completely the damage. And if we increase this sensitivity by adding PARP inhibitors, so by blocking the only way to repair the damage, the cells can only die. And this was the good rationale behind PARP inhibitors. And then there was the concept of the synthetic lethality, which means that you can induce to damage and block the way of repair by using different compounds, and in this way you cause the death of the cells. And then the first study with PARP inhibitors were done during the consolidation phase of the treatment, because today we know that in ovarian cancer the clinical history is set up by different steps, and we should be able to provide the right treatment for each step of the clinical course. And one of the big issues in ovarian cancer was, once I have achieved the response, how can I maintain it? And there were no good data with consolidation. And the point is that the first studies, which were done with the PARP inhibitors, single agent, they were only in patients with the BRCA mutated, because it was known that they were effective in this particular dysfunction of the cells, and during the consolidation treatment. And it was done only in the patients who were sensitive to platinum, which was already a good way to screen the right patients who can respond to the treatment. And after that, because of the good results, which significantly improve the duration of the response, PARP inhibitors were tested for the consolidation in first-line treatment. And we have already good results in a quite long follow-up showing that there's an improvement in the progression-free survival. So the point is that this is what is called the SOLO-1 study, a practice-changing study, because today we have two new important changes in the management of high-grade serous. First, all the patients at the diagnosis should be screened for BRCA mutation, either germline or somatic, regardless family history of previous breast or ovarian cancer. Second, the consolidation after the first line in patients who responded to the first line should include PARP inhibitor in patients with the BRCA mutation, and possibly with also in patients with some dysfunction not so well defined in this homologous recombination system. So it was really a successful story. It hasn't been so successful in other tumor types because the value, the relevance of BRCA mutation and the DNA repair system is not so high in other tumor types as it is in ovarian cancer. And then the point is that I discussed, which could be the future questions, which are the open questions. It's something we discussed already at the ESO Observatory, which was done in the ESGO Athens meeting last year. So you have good results, you open the use of new compounds, and obviously you have new questions because it's a continuous improvement. And the question is that what can happen if I use PARP inhibitor in first line? How can I affect the subsequent treatment of the patients? Or which will be the results of the studies which are already on long term, so the second line maintenance? And will be able to cure ovarian cancer or to increase the chance of the complete response rate and the cure of these patients? And still we don't have all the answers to that. So the point is that we should work together and we should try to exploit more these very fantastic molecules. And one potential way could be to combine two other tailored treatments. So there are already studies ongoing with anti-angiogenics, because we know that VEGF is very important for the microenvironment and for the immunotherapy of ovarian cancer. We know that it's decreasing the immunotherapy response. It's affecting the microenvironment, and it's increasing the evasion capacity of ovarian cancer. So perhaps by putting together anti-angiogenics and PARP inhibitors, we can get better results, and there are already studies ongoing. And also the other point is that there are really good rationale to combine PARP inhibitors with the checkpoint inhibitors, based again on biological evidence. And unfortunately, so far, the results with immunotherapy in ovarian cancer, even though the preclinical data were really good, and they started already in 2003, 2004 with the data of George Kukos, the point is that the results so far has been, let's say, disappointing. And perhaps that was also related to the kind of study which were done, because for a successful clinical development, you need not only the good preclinical rationales, but also a wise clinical development, to think which are the right study to be done. And so there's a lot of work ongoing, but clearly the situation is not comparable to what has been already achieved in cervical cancer and endometrial cancer, but definitely with other tumor types. And I think one important point as regards immunotherapy is that we cannot apply the same criteria, let's say immunotherapy or biomarkers, in all the different tumor types, because immunotherapy, it's something which is related to many different biological factors, and these biological factors might be different from one tumor to the other. For example, we know that the mutational burden of the tumor is very important for immunotherapy. For example, in melanoma is very high, as well in kidney, and also a bit in endometrial, but in ovary is very low. So perhaps this is one potential causes of this lower activity of immunotherapy. But anyway, there are good preclinical rationales, so that the work is in progress.